Research 1. I am Sir Jelby, your virtual research teacher.
Welcome to another topic in research entitled Methods of Experimental Research. I am Mark Angelbert Bidulay, and you can find me at JelbyPH. Plant identification. Plant identification is simply the determination of the similarities or differences between two elements, such as two elements are the same or they are different. The comparison of an unknown plant with a named specimen and the determination that the two elements are the same also involves classification. The information stored in classification systems becomes available and applicable to the material at hand. Plant collection sets. Always collect samples in an area with sufficient plant material. Ensure that the plant material is free from moisture and dirt. Arrange the plant specimens diagonally. Label the dry specimens. Plant material can now be submitted to Bureau of Plant Industry, University of the Philippines, or National Museum for expert plant identification and certification. Extraction. Extraction is the first step to separate the desired natural products from the raw materials. Extraction methods include solvent extraction, distillation method, pressing, and sublimation according to the extraction principle. Solvent extraction is the most widely used method. The extraction of natural products progresses through the following stages. 1. The solvent penetrates into the solid matrix. 2. The solute dissolves in the solvents. 3. The solute is diffused out of the solid matrix. And 4. The extracted solutes are collected. Phytochemical analysis or phytochemical screening are preliminary tests conducted to detect the presence of both primary and secondary metabolites in an extract. The Kirby-Bauer test. This is also known as the diffusion method. It is the most widely used antibiotic susceptibility test in determining what choice of antibiotics should be used when treating an infection. This method relies on the inhibition of bacterial growth measured under standard conditions. The CAM assay. The chorioallantoic membrane assay provides a simple and versatile animal model for the study of tumor formation, angiogenesis, and metastasis. There has been growing interest in this model as seen by the dramatic increase in the number of publications regarding this model. Brine shrimp lethality assay. It is the most convenient system for monitoring the biological activities of various plant species. This method is very useful for preliminary assessment of the toxicity of the plant extracts. Rapidness, simplicity, and low requirements are several advantages of this assay. Common blood test. The common blood tests are the following. Complete blood count. The most common test is a complete blood count that analyzes the numbers and appearances of blood cells. Hematocrit or HCT. This test measures the percentage of red blood cells to detect anemia and hydration. Hemoglobin and mean corpuscular hemoglobin concentration. These are the oxygen-carrying pigments of red blood cells. White blood cell count. This test measures the body's immune cells. Increases or decreases in the WBC indicates certain diseases or infections. Granulocytes and lymphocytes monocytes. These are the specific types of white blood cells. Platelet count. This test measures cells that form blood clots. Albumin. This is a serum protein that helps evaluate hydration, hemorrhage, and intestinal, liver, and kidney disease. Alanine aminotransferase. This test may determine active liver damage. Amylase. Elevation in this test indicates pancreatitis or kidney disease. Aspartate aminotransferase. Increases in this test may indicate liver, heart, or skeletal muscle damage. Blood urea nitrogen. This test determines kidney function. An increased level is called azotemia and can be caused by kidney, liver, and heart disease, as well as urethral obstruction, shock, or dehydration. Creatinine. This test reveals kidney function and helps distinguish between kidney and non-kidney cause of elevated BUN. Glucose. Glucose is a blood sugar. Elevated levels may indicate diabetes mellitus. Low levels can cause collapse, seizures, or coma.
That's all for this day. If you have any questions, follow me at the following. Thank you.